What if I made fettuccine Alfredo and then decided what I really wanted was cheesecake? Could I turn one into the other? Well, maybe I could if I could turn back time and go back to the original ingredients. Then I could make something else entirely. The idea of turning back the clock and rebuilding something from scratch is the basic premise behind stem cell research, which aims to transform one kind of cell into another. Correspondent Chad Cohen reports, recent groundbreaking discoveries seem to be bringing them that much closer to their goal.
It feels like a lot of people are stabbing me with knives from the inside of my body, all the way down to my bone. Sometimes I couldn't catch my breath. It was so intense.
I'm in tears, basically. It's just pain everywhere. It's going through your head, your back, your legs. You can't do anything about it.
Stephanie Termitus and Emil Reed know the inside of Children's Hospital Boston better than any teenager should. They both suffer from sickle cell anemia. Instead of the plump, donut-shaped blood cells most of us have delivering oxygen to our organs, their blood cells are sickle-shaped. Banana, as Stephanie likes to say. These cells can't deliver oxygen very well, which causes the kids excruciating pain. Emil's mom is on a first-name basis with the hospital staff.
They've given Emil pain medication, and it doesn't take the pain completely away. It sort of numbs it. I mean, that's happened where he's ended up in the intensive care unit for 4 months. So it's pretty serious.
The misshapen, stiff red blood cells get sludged and clogged into the blood vessels that feed their organs and their muscles and their bones, and this is intensely painful.
It's been 50 years since researchers found the cause for those misshapen blood cells. A single change in a single faulty gene. Sickle cell was the first genetic disease ever identified.
Give me a big deep breath.
Yet despite decades of searching for cures, all we can do is treat the symptoms, or in rare cases, perform bone marrow transplants, which are dangerous and carry a high risk of rejection.
And so this is a condition that we need a new approach for.
Lately, that new approach has involved embryonic stem cells, cells that are pluripotent, meaning they can grow into just about any cell in the body. When they were discovered more than a decade ago, it was thought that stem cells could fix not just sickle cells, but the damaged cells of countless diseases: Parkinson's, diabetes, ALS. That's what's driven George Daly, who also heads a stem cell lab right across the street from his patients.
Thinking about the potential that this has for changing the way that we not only study disease, but one day treat disease is really very, very exciting.
But there's a problem. Stem cells, for the most part, come from human embryos, from that time just after sperm meets egg, when we're made up of just a few dozen cells, and the function of those cells has yet to be determined. The main source for the embryos are IVF clinics, where surplus embryos are often discarded as medical waste. Still, harvesting the stem cells destroys the embryo, and for many, that's morally wrong. Others believe that holding back medical progress is also wrong.
Here we are at the dawn of this whole new field, all this excitement, all this possibility, and yet we're working with one hand tied behind our back.
But in 2007, some experiments were conducted that many believe will finally bring the fighting to an end. Japanese researcher Shinya Yamanaka figured out a way to take an ordinary skin cell from an adult, turn back its genetic clock, and transform it into the equivalent of an embryonic stem cell. No embryos required. Yamanaka's motivation came when he first glimpsed human embryos under a microscope about 10 years ago.
I have 2 daughters, and I thought the differences between those small embryos and my own daughters are very small.
The realization presented some conflict for him, since as a physician, he believed that embryonic stem cells were his best shot at treating disease.
To me, treating patients or saving patients is the most important thing to do. But if we can avoid the usage of human embryos, we should avoid.
Ironically, Yamanaka had to use embryonic stem cells in order to find a way to do without them. He started by exploring one of their fundamental properties. Virtually every cell in the human body has the same DNA. Heart cells, liver cells, skin cells, all share the same 20,000 genes. During our development as embryos, though, different genes in different cells get switched on and off in different ways, and that's what creates all the different types of cells in our bodies. It's called cell programming. Yamanaka believed if he could find the gene switches responsible for programming stem cells, he could flip those same switches in adult cells, like skin cells, and reprogram them back to the moment before their destinies were determined.
Each cell has at least 20,000 genes. So that means we have to find those important switches from the 20,000 candidates.
With so many genes to choose from, so many potential combinations, the search could have been infinitely complex.
Yamanaka's insight was to appreciate that it was a very limited set of genes, and he set out to identify them.
First, he reduced 20,000 possible gene candidates down to 100 using online databases. But then the work got harder.
We spent like 3 years to study the function of those 100 genes.
Were there people saying, give up, there's no use in this?
Yeah, many people told me that this is going to be very difficult. You will fail.
Using specially engineered mice called knockouts, he tested each gene's ability to make pluripotent stem cells, eliminating them one by one. After more than 3 years culturing hundreds of thousands of cells, Yamanaka narrowed the gene pool down to 24 genes, and finally 4. Then came the moment of truth, getting these 4 painstakingly selected genes to make stem cells. He took some skin cells from an adult mouse, then used a virus to insert the 4 genes inside them. 2 weeks later, the skin cells in the petri dish had completely transformed.
We saw cells which looked like stem cells. So it was the moment, we were very, very excited and we were very surprised.
Yamanaka dubbed the cells induced pluripotent stem cells, or iPS cells, and found they were virtually indistinguishable from embryonic stem cells.
I can't see a difference. I wouldn't expect to be able to see a difference, but
No, we can't see differences either. So these embryonic stem cells and induced pluripotent stem cells are indistinguishable. They are the same cells.
it's amazing.
Yamanaka's experiment was bold. Some might say foolhardy. I think it's the type of experiment that would be laughed out of the room in a standard peer review study section. Would have never gotten your grant funded with that experiment.
Really?
And now it's probably going to win Shinya Yamanaka a Nobel Prize.
Creating stem cells without an embryo in mice certainly made headlines in the scientific community. But less than a year later came the news that caught the world's attention. Based on Yamanaka's work, 3 independent scientists, James Thompson in Wisconsin, George Daly in Boston, and Yamanaka himself, transformed human skin cells into iPS stem cells. It was a monumental breakthrough. And in George Daly's case, the doctor even experimented on himself.
So what we're going to be doing is just obtaining a small biopsy of the skin.
We have a protocol where we can have anyone walk in, roll up their sleeve. We take a very small skin biopsy, smaller than the eraser at the end of a pencil.
And we'll just snip this and we are good to go.
Fantastic. And those skin cells are then put right into a petri dish, and then within a week, all of a sudden, this huge bloom of cells appears. And then you bring into them the 3 or 4 genes that do the reprogramming. What's really remarkable is that just simply putting those genes into the cell and making them work starts this whole process. It takes those stable specialized skin cells and erases all the skin functions and reactivates, enlivens the embryonic functions and turns that skin cell back into a pluripotent embryonic cell.
Back in time, basically.
It's back in time. I mean, it's like a whole altered universe. I mean, it's really changed the fundamental nature of that cell. How many times would you say you've been in the hospital in your whole life?
I can't even say it's so many, like 10 times a year.
So how long before this technology actually helps patients like Stephanie? Well, some serious challenges will have to be overcome first. For one thing, that virus to shuttle Yamanaka's 4 genes into adult cells can mutate a patient's DNA and cause cancer. At least one of the 4 genes is an actual oncogene. It definitely causes cancer. And a high percentage of the mice created with these stem cells did develop cancer. But the promise of these cells far outweighs their problems. And as we speak, researchers around the world are figuring out how to safely use them. Late in 2007, Rudolf Janisch of the Whitehead Institute at MIT demonstrated a powerful application of iPS cells. He used them to cure sickle cell anemia in mice. To do it, he first had to give the mice the disease.
So you're able to basically give the mice sickle cell disease and use that as a model?
Yes, these mice were highly anemic. They had just stopped growing. They were very rather small. They wouldn't gain weight.
Sure.
They would die early. I mean, it's a very faithful model of this major human disease. You would take then a skin cell from this mouse and reprogram it to iPS cells.
Janisch made the stem cells using Yamanaka's same 4 gene switches. But this time, he removed that nasty oncogene once it had done its job.
So now these iPS cells, they didn't need it anymore. They didn't have that oncogene. So that was useful. And then the next thing was we repaired the genetic defect by gene targeting.
Janisch targeted that single sickle cell mutation, fixed it, then prompted the stem cells to become blood cells and injected them back into the mice. Since these cells came from the very same mice, they're a perfect match. There was no chance of rejection.
And to our really delight, the blood of the mouse totally normalized, and they began to gain weight. They have lived, as far as we know, they have no problem. So they're totally cured, these mice, from the sickle cell condition.
So, have these new stem cells made embryonic stem cells obsolete? Well, until we know for sure whether they can faithfully grow into all the different cell types, the answer is definitely no.
I'm not willing to concede that iPS cells will ever fully replace human embryonic stem cells. The embryonic stem cell line remains the gold standard. Okay, can you take a big deep breath? It has been the most exciting past few years I could imagine in my career. And I hope that it is translated into real treatments for my kids.
My favorite subject is science.
That's good. That's very good. We like to hear that.
The fact that they can take your own skin and turn it into something that can cure a disease like that, it's incredible.